Functional genomics investigates how genes and non-coding elements within a genome contribute to biological function, phenotypes, and disease. Rather than cataloging sequences, it focuses on dynamic activities, when and where genes are expressed, how regulatory elements modulate transcription, and how genetic variation alters molecular networks. By linking genotype to function, functional genomics translates static DNA information into mechanistic insight about cells, tissues, and organisms. Key experimental approaches measure different layers of gene activity. Transcriptome profiling (RNA-seq) quantifies gene and isoform level expression across conditions and cell types. Chromatin assays such as ATAC-seq and DNase-seq map regulatory accessibility, while CHIP-seq identifies binding sites of transcription factors and histone modifications that mark enhancers and promoters. Proteomics captures protein abundance and post-translational modifications, and single-cell methods scRNA-seq, SCATAC-seq resolve heterogeneity and developmental trajectories. Perturbation techniques, CRISPR-based knockout, interference, activation, and pooled genetic screens test causal roles of genes and non-coding elements at scale. Integration of these data types is central to functional interpretation. Correlating chromatin state with expression identifies candidate regulatory elements. Linking genetic variants from genome-wide association studies (GWAS) to regulatory maps helps prioritize causal loci. Combining perturbation outcomes with transcriptomics reveals downstream pathways and buffering interactions. Computational frameworks, co-expression networks, machine learning models, and causal inference tools organize high-dimensional data into testable hypotheses and predictive models of gene function. Applications span basic biology to translational research. In developmental biology, single-cell atlases map cell-type-specific programs and lineage decisions. In disease research, functional genomics pinpoints driver genes, interprets non-coding risk variants, and uncovers disease-associated regulatory circuits. Drug discovery benefits from target validation through genetic screens and from expression signatures that predict compound mechanisms or off-target effects. Agricultural and environmental genomics use similar principles to optimize traits and understand organismal responses to stress. Despite rapid advances, challenges remain. Many regulatory elements act in context-dependent manners that are difficult to assay in vitro. Linking distal enhancers to their target genes across cell types is still imperfect, and functional effects of rare or complex variants can be subtle and polygenic. Data integration is computationally demanding and sensitive to batch effects and annotation biases. Ethical considerations arise when applying functional genomics to human germline editing or predictive models with clinical implications. Looking ahead, continued innovation in multiplexed perturbations, improved single-cell multiomics, spatially resolved assays, and interpretable machine learning will deepen mechanistic understanding and improve translation. As atlases expand across individuals, developmental stages, and environmental contexts, functional genomics will increasingly enable precise mapping from genotype to phenotype, guiding both scientific discovery and clinical decision-making.